Okay, so topic today, eubacteria and archaea. Notice that I said archaea and not what? Archaea bacteria. They don't call them that anymore, and we'll get to why in a minute. They dropped the bacteria part for a reason. But EU means what? True. These are your normal, everyday, regular bacteria. We talked about most of this before, so I'm going to review it, but I'm going to move pretty fast in this part. We know they're prokaryotic. Literally, that word prokaryotic means what? Before the nucleus. Because supposedly, prokaryotes came first, and a bigger one took in a smaller one, several smaller ones, and that whole idea is called what? Endosymbiotic theory. And that over time, the smaller ones evolved to become what? Yeah, but the smaller ones inside the bigger one became organelles. That's the general idea. Another thing that you had a question about on your test, all prokaryotes do have cell walls. Eubacteria have cell walls made of a particular kind of chemical called peptidoglycan. Part sugar and part protein. The job of the cell walls, most cell walls, their main job is to keep the cells from what? Bursting when they're in what kind of solution? Hypotonic, right? Less solids, water would go in and make them swell up. Another thing about bacteria that was kind of related to your test, they don't have any membrane-bound organelles. They do have what? Ribosomes, which are not what? Membrane-bound organelles, right? Did everybody sign the sheet? No. Okay, that's over there. Remember that ribosomes make proteins. What's the advantage of having organelles?
Please go to your office.
Specialization and therefore the division of what? Work. What's the more common phrase? Instead of division of work, we call it division of labor. Divide up the work, become really good at doing specific things. Bacteria can't do that because they're one piece. We know they don't have a true nucleus. Their DNA is just floating out in the middle, and sometimes it's called the nucleoid because of that. How much DNA do they have? One circular chromosome. Plus, sometimes they have some really tiny extra pieces of DNA called what? Speak up. Plasmids. And those are important because what can they do with those little extra pieces of DNA sometimes? They can exchange. They hook together, they conjugate, and they swap them. And that's sort of like bacteria having what? Sex. What's the purpose of sex? Create genetic variation. Create new organisms with different combinations of genes. When that happens in humans, obviously it makes a new individual. Here, start with two, you still have two, but the two that come out of conjugation are different than the two that went into conjugation, right? So it's mixing up the genes. And remember the way this conjugation idea works. You got two bacteria. They hook themselves together with what structures? The pili. They have these little structures called pili. They're sort of like cilia. Most of the time, what do pili do? What's their major function? They're like, I want you to think of them like bacterial Velcro. They help them stick to things. But they hook together with specialized pili. This would be like their main chromosome. And maybe these little loops here would be their plasmids. And when they hook together, they can swap plasmids. And therefore swap genes and create bacteria with different combinations of genetics, different combinations of genes. That's conjugation. Why is that not going away? Most of the time they reproduce by prokaryotic fission, binary fission, which is what kind of reproduction? Asexual. And we talked about this before. When does asexual reproduction make sense?
When your environment is good.
The environment is stable, and you're good at living in that environment, right? You've adapted to that environment. It's staying the same. Doesn't make change. If it ain't broke, don't fix it, right? That's a common old folks saying, if it ain't broke, don't fix it. That's when asexual reproduction makes sense. When the environment's stable and the organism is good at living in that environment. It's fast, right? But what does it not do that sexual reproduction does do? It doesn't create those new combinations of genes. It doesn't create genetic variation. So sexual reproduction makes more sense when the environment is like changing, and therefore you're able to what? Evolve. The only way we can evolve is if we have different combinations of genes. It's about evolution. Now, another thing I'll mention here about bacteria, we've talked about their shapes before. They come in three main shapes. What are they?
Coccus, bacillus, and
Yeah. Coccus, which is what kind of shape? Yeah, but let's be a little bit more exact here. Sphere shape. A bacillus is kind of like a capsule, a ride, and a spirillum is just what it says, spiral. Okay. You're okay. Another thing about bacteria that's kind of unique to the eubacteria. Here's this little circular chromosome. It's in an environment that is getting bad. Maybe it's getting cold. Maybe it's getting acidic. Maybe it's getting hot. Something's going wrong with the environment. So maybe salty. Something's happening with the environment. The bacteria, it doesn't think the way we think. But what does it do to that chromosome before it dies? It seals it off in a coat. It makes this hard coat around it. And the cell dies. But the DNA is preserved. And when the environment gets better, what happens? Yeah, that is the endospore. The DNA in a protective case is the endospore. But when the environment gets better, the spore goes away and the DNA does what? Forms a new cell. So it makes bacteria hard to kill. Bacteria that can do this are hard to get rid of. Yeah.
the DNA form a new cell, like out of nothing, or it still some stuff in there?
Well, there's still some things in there, and then they'll take things from the environment to get what it needs. But most of the cell dies. You might think of an endospore sort of like what other structure in biology? It's kind of resistant to weather problems. Seed. It's sort of like a seed. I mean, plants, seeds can dry out. You put them in the freezer, soak them in water. You can do a lot of things to a seed, right? And they're still okay. That's sort of what an endospore is for a bacteria. It's more resistant than a seed, though. Seeds can only take so much and they die. Endospores help them deal with bad environments. We talked the last time about a lot of diseases caused by bacteria. And there's a short list. Some other things that we might put in there: the bubonic plague, the Black Death, TB. All those things would be bacteria. A long time ago, back close to the end of World War II, what did doctors think about bacterial diseases? End of World War II, maybe 5 years later. They thought we were done with bacterial diseases. We had conquered them. It was over for bacteria. Why? Antibiotics were brand new. Penicillin was new. And penicillin killed just about any bacterial infection at the time. But what has happened? They have evolved. And bacteria evolve really fast. Way faster than we evolved. How come? Every time they reproduce, there's a chance that what will happen? They'll mutate. And mutations lead to new what? New genes, new traits. And the thing about bacteria, how often do they reproduce? Yeah, really, really fast. Sometimes every 30 minutes. So things that have a short life cycle, things that reproduce fast, evolve fast. That's why we have a hard time killing certain kinds of bacteria with antibiotics. We get a new antibiotic, and they evolve to be resistant to it. With MRSA, staph infection, TB. There are very few antibiotics that will kill tuberculosis anymore. What else in nature evolves really, really fast? And that makes them hard for us to handle. Well, viruses. Viruses are kind of tricky though because they're really not alive. But true. That's not what I'm talking about. What living thing? Because they reproduce so fast and they have short.
Worms?
Well, rabbits is not what I was thinking. I was thinking insects. We're constantly having to develop new kinds of pesticides to get rid of them, right? Because they evolved to be resistant to them. Anyway, we talk about all these bad things bacteria do, but by far, most of the things they do are good things. Their most important role in nature, we've mentioned before, is decomposition. What we might call mineral what? Mineral recycling. They break down dead things, and the minerals that are in those dead things end up back in the soil, so other things can use them. A test question, that if you were at the last study session, you got a preview right before the test. Another important role of bacteria is nitrogen fixation. There is plenty of nitrogen on Earth, right? But there's not, in a lot of cases, there's not enough available to living things, because where's most of our nitrogen locked up at? In the air, as N2, which has got a triple covalent bond, which is almost impossible to break. There are very few things that can break it. Because we can't break that, we can't use that. Most things can't use N2. But there are a few things, mostly what, that can break that bond? Mostly bacteria. Bacteria that tend to live where?
In the grass.
Yeah, usually on the roots of plants in some kind of symbiotic relationship. And they take that N2 and turn it into what? Yep, ammonium ion, NH4, or sometimes nitrate ion. Either way, those are forms of nitrogen that plants and animals can use, right? So the nitrogen that we use tends to come from bacteria in that process of taking N2 to ammonia or nitrate, that process is called what? Nitrogen fixation. All right, so let's move on for a minute. Still haven't quite figured out this new piece of software they put on here.
And the plant sees the ammonium nitrate.
Yep. And then a lot of times animals will get it from eating the plants, or they'll get it from the dead, from the soil, from the plants. Either way. Yeah.
So how does bacteria break it down?
They have some enzymes that we don't, that'll allow them to do it. What'd you say? Nitrogenase. Yeah.
Do the plants let it back into the air feed? Or
No. And it does. A lot of it does end up back in the air because there are other bacteria that make nitrogen go the opposite way. They'll take nitrate or ammonium and turn it back into N2. So it's a cycle. That's what's called a nitrogen cycle. It goes back and forth. Here's a picture of a general eubacteria. Some have flagella. Not all of them do. Some have them for movement. Most of these things we see on here are pili. It's got the one big chromosome. Actually, big is a misnomer, because compared to just one of your chromosomes, how big is that? Tiny, tiny, tiny. Compared to even one of your chromosomes, that's really small. Here's a plasmid. They might have several plasmids. Those are, remember, extra loops that can be swapped during conjugation. They've got ribosomes. They're all free-floating, which means typically bacteria are going to make proteins that are going to remain where? In the cell. Because in us, that's what our free-floating ribosomes do, right? If we want to send something out, those ribosomes are going to be where? On the rough ER. Exactly. Also remember that bacteria, they all have a cell membrane. And up here, I think that's the blue part. Yep, the blue part's the plasma membrane. And then outside the plasma membrane is the wall, which that in bacteria is made out of peptidoglycan. And some bacteria on top of that wall, on the very outermost part, we'll just draw it in here. What might my black drawing there represent? Do I know? A mucus coat called the capsule, or sometimes it might be called the glycocalyx. But that's there mostly not to keep water out, but to what? Keep it in, to keep it from dehydrating, keep it from desiccating. This is showing you binary fission or prokaryotic fission. Before it ever starts, what has to happen? DNA had to be copied, right? And that's what we see here. One chromosome, now we got two. Copy the DNA, replication. That one chromosome tends to attach to, what's it attaching to up there?
Membrane.
Cell membrane. And the cell grows. And as it grows, those two chromosomes get pulled apart. And then it starts to what? What's happening right there? What's that called? Invagination. Starts to pinch. Now, bacteria have one thing to worry about that we don't, and that's what? What kind of complicates this in bacteria and fungi, plants? Well, true, but they've got not only got to split the cell membrane, they got to build a new wall down the middle, right? That becomes a little complicated when they have to build a new wall between those two cells. And we'll get in later how that happens, but they do have to build one side of a wall on each new cell. Question?
So chromosome attached to the membrane.
Membrane grows,
This
chromosomes get pulled apart, cell invaginates. There are those shapes we were talking about earlier. Spirillum, spiral-shaped bacteria. Bacillus, they're shaped like a capsule or rod. And this is what we call coccus, but this has a special name. Anybody know what a chain of spheres is called? It's called streptococcus. Strepto, when you have strep throat, you have this kind of bacteria that grow in chains of spheres. Streptococcus. What's staphylo mean? Staphylococcus. On staph infection kinds of stuff. That means the bacteria are arranged like this.
Hey, that's not saying.
Like a cluster.
What's that?
That's staph.
Ooh.
Now, we haven't mentioned this one in here before, I don't think. Cyanobacteria. What does cyan mean? What color? It might be a little more blue than Casey's shirt, but that's pretty close. Okay? That's pretty close to cyan. Maybe a little bit more bluish, but bluish green. Yeah, I'll say maybe it's a little more bluish. But regardless, these are bacteria that can do what? What can they do? They can do photosynthesis. There are bacteria that can do photosynthesis without having what? Without having chloroplasts. But they do have what? What's the green? Chlorophyll. And they don't, that says a thylakoid. I know they don't really have thylakoids though. The chlorophyll to work has to be embedded in what? In a chloroplast. Where's the chlorophyll?
membrane.
It's embedded in the membrane of the thylakoid. It's a protein, right? It's got to be in a membrane to work. What's the only membrane that bacteria have? Cell membrane. So their chlorophyll is embedded in the cell membrane. What you're seeing here is that what's going on with the cell membrane? It's all what?
Square.
Well, that's the wall. What's going on in here where that green's at? It's all bent up, twisted in order to increase surface area.
Nice.
But the chlorophyll has to be embedded in the membrane, and that's what we're seeing there. For years, these weren't called cyanobacteria at all. Anybody know what they used to call them? Blue-green
Bacteria.
algae. They don't call it that anymore because they figured out they're not what? They're not algae. Because algae are made of what kind of cells? eukaryotic cells. Okay? These are not. There's no nuclear envelope. There's no organelles. That's why they're bacteria. That's why they changed their name to cyanobacteria. They're not eukaryotes like other algae are. But they can do photosynthesis. Important part of the food chain. It'd be hard to live without. Gotta figure out how to set this thing up. Let's talk a little bit about archaea. Now, for a long time, why did they name them archaea, first of all? What's archaea mean? It means old. For a long time, scientists thought what about these things? They were the first, not just bacteria, but the first living things on Earth. That's not what they think anymore. So the name doesn't really mean what it should mean anymore. They think that normal eubacteria probably came first, and that the archaea and the eukaryotes branched off of that. And I'll show you a tree that kind of gives you an idea of how that happened in a minute. But for years, all bacteria and all archaea were classified in what kingdom? This may be before your time in biology. For a long time, they were in a kingdom called Monera. Heard of that before? Yeah, for a long time, that's all the bacteria were in that kingdom, archaea and bacteria. But what is our system of classification based on today? Why are you in the same group as, what order do we belong to as humans? The biological order. Let's just go through human classification for a minute. What is the biggest group in our classification system?
Domain.
The domain.
Isn't there a new one though?
Well, that's the domain. Domain's the new one. What domain do we belong to? Eukarya. All eukaryotic organisms belong in that domain. All right, what comes next? Kingdom. We are in animal kingdom. Animalia. What comes next? You have a phylum, right? And our phylum is called Chordata. And we'll learn more detail about this later, but essentially, what does it mean to be a chordate? You're a vertebrate. Essentially means you're a vertebrate. All the vertebrates and a couple other things belong in that phylum. For right now, vertebrates. What comes next? Class. Our class is Mammalia. And then after class comes order, and our order is primates, primata, Latinized names. What comes next? Family. And what's our family? Hominidae, which are hominids, it's sometimes just called hominids. Our genus is Homo, and our species, obviously, sapiens. Now, two things to know about this setup. You cannot be a mammal without also being what? A chordate and an animal and a eukaryote, right? As you go down this list, to be in the same order with something, you also have to be the same class, the same phylum, the same kingdom, the same domain. As we go down this list, what's happening to the relationships between things in these groups? They're getting more related. The groups are getting smaller, right? More specific. And the things in the groups are more and more related. What else is in this primate order with us? Monkeys, gorillas, orangutans, chimps. Why are we in the same order as those things, according to biologists, modern biologists? What's it all based on?
DNA.
It's all based on an idea called phylogenetics. That's a word you need to know. And basically what it means is that it's based on genetic and what kind of relationships? Genetics and evolutionary relationships. Taxonomy today is based on those two things, genetics and evolution. So you're in the same group as primates, as other primates, because you share a lot of DNA with those primates. Sometimes 95% with certain species of monkey, gorilla, Asian kinds of things. And scientists would mean because of that, that must mean what? You're related through evolution to them, the whole conserved properties deal. So back to what I was saying here. For years, archaea and eubacteria were classified in the same domain. Well, same kingdom, Monera. What caused scientists to split them up? Because they do look a lot alike. Why'd they split them? Related to our discussion over here. They started looking closely at their DNA sequences. And what did they find about their DNA sequences? They're pretty different. And they also found that archaea DNA was actually more like what? Than it was bacteria DNA. It was more like eukaryotic DNA. They actually think archaea are more closely related to eukaryotes than they are to other prokaryotes because of the DNA sequences. Because the whole system, remember, is based on this, genetics and evolution. Now, the one thing you probably might remember from regular biology about archaea, what did Miss Kenny teach you about?
They live in harsh environments.
They live in harsh environments. I'm going to tell you, forget that. Because it's not true, okay? The first ones they found and they realized they were something different, that's where they were living. They do live in those places. But guess where they also live? Pretty much everywhere else, okay? They found them in human intestine, they found them in oceans and ponds and lakes and streams and everywhere in between. For a long time they thought they only lived in these extreme places. Not true. They live all over the place. But in terms of why they separated them and made a whole new domain for them, right? How many domains are there? 3. Eukarya, eubacteria, and archaea. There's 3 domains. And those all get divided into separate kingdoms. But why they did, first of all, is they found they're chemically distinct from other bacteria. The DNA, let's start with that. Their DNA, a lot of their genes are more similar to eukaryotic genes than they are to bacteria genes. Which to biologists means what? They're more closely related through evolution to eukaryotes than they are to other prokaryotes. Some other things that are different, their cell walls are made differently. Archaea have cell walls, but they're not made out of what? They're peptidoglycans. They're kind of variable in terms of what they're made out of. I'm not going to give you one specific thing and say archaea bacteria cell walls are made out of this, because they're made out of different things. They're not made out of peptidoglycan. A couple of other things. When we talked about our DNA, we talked about how when it comes time to do mitosis or meiosis, eukaryotic DNA wraps around what to form chromosomes.
Protein.
What kind of protein? What's the name for the kind of protein that wraps around? Wraps around these proteins called histones. And they're sort of like spools to organize our DNA. Eukaryotes have histone proteins that spool up our DNA. Eubacteria don't have those. Archaea bacteria do have them. So that puts them more related to us and less related to regular bacteria. They have histones. Another thing that sort of puts them closer to us is their the enzymes that they use to do replication and transcription and translation. Those enzymes are much more like what than what? Their enzymes for replication and transcription and translation are much more like the eukaryotic version of those enzymes than they are the bacteria version of those enzymes. Which means what else has to be more similar? If the enzymes are more similar, what else has to be more similar? The DNA, because enzymes are what? Proteins, which are coded for by DNA. So there's a lot of genetic similarities between archaea bacteria and eubacteria. Eukaryotes, I'm sorry, thank you. Between archaea bacteria and eukaryotes. So remember, when you hear about archaea, don't just think extreme environments. Yeah, they're found there, but they're found in other places. Make sure you realize that they're different from other bacteria genetically. They seem to be more related to eukaryotes than they are to prokaryotes, other prokaryotes. We tend to divide them up into 3 groups. And this is being changed as we're finding more and more of them. But for a long time, one group is called methanogens. That means they what? Not just found in methane-rich environments, they what? They create the methane. They give it off. They're gassy little things. Thermoacidophiles.
They're an acidic element to create acidic.
No, you had it right the first time. They like not only acidic places, but they also like hot, right? Phile means what? Love. Like pedophile, right? That's a nasty word. Yeah. You'll remember that, right? Phile means to love. Okay? And then extreme halophiles like to where? What kind of extreme places?
Hell.
What does halo mean? Like halogen.
Like really high up.
What's halogen mean?
Yes.
Mm-mm.
Really?
Joseph Crandall Troy, please come to the front office.
Halo means salty.
Really?
Because most salts have halogen in them, right? A lot of salts anyway do. Salt formers. So these are bacteria that like really salty places. What would be an obvious place for those?
Ocean.
No, more salty than the ocean.
You said an obvious place.
Yeah, like the Dead Sea, which may not be obvious to some of them, but it's obvious to Jeff. Okay? The Dead Sea is dead because what? It's so salty, not hardly anything can live there. These things can, but not much else. Don't change that.
So the thermoacidophiles, they love the hot and acidic places.
Mhm.
And how do they create methane? The methane.
They're well, you created too. And that's what natural gas is, right? So it's a waste product of metabolism. Where might you find thermoacidophiles? Like deep ocean vents, Yellowstone maybe. You got a lot of those geysers and things like that. Yellowstone. Did I say that incorrectly? Yellowstone. There we go. Roll Tide. Anyway, that was a redneck joke, by the way. Archaea. Most of this we've already said, but just to stress it. Classified in their own domain. Archaea is the name of that domain. No longer called archaebacteria because they've been found to be what? Genetically distinct from other bacteria. They're different. And like we said, they are found in extreme environments, but they're also found in not so extreme environments. So that whole extreme environment argument doesn't always work anymore. Just the last couple years. The classification system changes every day pretty much. As they learn more about DNA and how one thing's related to another, that whole classification system changes. Yes?
Is bacteria basically the same thing as prokaryotes?
Well, archaea and eubacteria are prokaryotes because neither one of them have a membrane-bound nucleus. So they're both prokaryotes. All right, now in terms of things that are similar to them in normal, regular old eubacteria, they're both single-celled organisms. Their size and shape are very similar to regular eubacteria. They don't have a membrane-bound nucleus. What do we call their DNA? Since it's just floating around, it's called the nucleoid, not the nucleolus, nucleoid. They also don't have any membrane-bound organelles. This part you may not know what it means. What are introns? My genetics folks know what introns are? All right, in us, there are sequences of DNA that don't typically get transcribed, which means they don't tend to do what? If they don't get transcribed, they don't get used to do what? Make proteins. Well, not necessarily recessives, but for a long time, scientists called this junk DNA. Because they didn't think it did anything. They thought we had more than half of our DNA that never did anything. Lately, they figured out they were wrong. It does do some things. But, I kind of misspoke there. These extra sequences of DNA, they do get transcribed into RNA, but before the protein gets made, what happens to those intron sequences? They get taken out, so they're never used to make proteins. Eukaryotes all have introns. And we'll talk a lot about introns later, so don't get bent out of shape about it right now. But bacteria don't, and neither do archaea. What that means is that that makes their DNA work in a lot more simplistic fashion than ours. Because before we can use our RNA, we have to do something called processing to it. And that essentially means taking out what? The intron sequences, so that it'll work. They don't have to do that because they don't have any introns to begin with. All their DNA is transcribed, it's all translated, it's all used to make protein. So it's a simpler system than ours. Again? Okay. It turns out what's treated as an intron in you one time through, in a different environment might what? In you, you've got these intron sequences that typically aren't used to make proteins. But let's say something changes in your body or in the environment. What might happen to that sequence that hasn't been used much? It may start getting used, and it gives us the ability for one gene to actually code for what? Multiple different proteins based on what? Based on the environment. You probably don't have time to do it today, buddy. I'm leaving at 4. Yeah. You can do it Friday. You can do it then.
So, introns are parts of DNA that are used.
That are typically not translated. They're typically not used to make a protein. But what that gives our DNA the ability to do, since we got these extra pieces of DNA, is that we can, one gene, depending on the environment, can actually make more than one kind of protein. So it gives us some variation. So again, I don't know that I would say backup, but it's like an emergency plan, yeah. If something weird's going on, now you make a different protein. Hopefully it'll help you do something better. All right, let me finish up by showing you a diagram. I gotta fix that.
Just move it over where.
Yeah. Anyway, this is supposed to be what's called a phylogenetic tree. You now know what phylogenetics means. It means what? How are things related through genetics and evolution? So this is supposed to be like an evolutionary tree of life. That's what it's supposed to be. So somewhere way back here, supposedly was the first living thing. Everywhere there's a branch, like right in there. That's where the common ancestor of everything over here and everything over there was. So whatever living thing was right there, in that little V, was the common ancestor of all modern life, right? Some descendants of that ancestor evolved this way to become what? Bacteria. Some evolved this way. Somewhere along the line, some relative of this thing changed. And some of its descendants became what? Archaea, and some of its descendants became eukarya. So right in here, that's the descendant of archaea and eubacteria, whatever that thing was. But what you ought to see is that this shows you that what two groups are more related. Which one's the odd man out? Bacteria, because these two groups, archaea and eubacteria, share this common ancestor, right? Eubacteria only share this common ancestor with all three. These two share, I mean, these might be like first cousins, and therefore they'd be what to this guy over here? If these two are first cousins, they're probably this guy's what? Second or third cousin, right? It's further down the family tree. But again, these are our 3 domains. And this diagram, which we will talk a lot more about as we go through the class, phylogenetic tree. How are things related through evolution and genetics? And that's a good place to stop.
I was so close on everything. So close.
Most of it's